Good evening, everyone. Am I audible? Okay. Am I audible and visible to all? Someone please confirm if they can hear and see me. We will wait for another 5-10 minutes for the people to join. Okay, so good evening everyone. I am Angira and I am a PMRF research scholar in Department of Chemical Engineering, IIT Bombay. So I am in my 4th year of my PhD and I have been given the responsibility to teach you all, or one second, I have to admit people as and when they come. So this live session is actually targeted towards the students. I think there is some audio problem coming. Is it fine for you all, or are you hearing a sharp noise? So from here on, actually I've joined from 2 devices, so I think that's a problem because my laptop camera is not working. So from here on I will be trying to teach or discuss without my camera, so that this disturbance of background noise is solved. Okay, so to start with, I hope everybody has looked at the online available lectures of Professor Gargi, and they know that they have been given assignments over the past few weeks, which you were asked to solve. So I would be discussing certain questions or certain part of the assignment which was in week 0, and certain parts of the assignment 1 also, so that you can get to know how this or the approach to solve certain questions like this. Okay, so here in this kind of live interaction you will get questions from the assignments. Some questions can be made by me also, and I will prefer everybody to take part and actually take part in the discussion, so that we can understand and discuss about the problems to understand the concepts better. Just for a quick surety, just let me know if I'm audible, because in my camera there is a problem, and therefore I'm obviously not visible. So just confirm if I'm audible. Am I audible? Okay, to start with, the first question that I would be discussing uh, is the following. This domain is characterized by ancient bacteria that can live in extreme environments such as volcanoes. So the options of this particular questions are archaea, bacteria, fungi and protozoa. So may I request one of you to actually tell me uh, which one of this will be the answer?
Archaea.
Yes. So the answer is actually archaea. Why so? Because uh, when the world developed, archaea was one of the primary organisms that developed in the primary world. So the primary world had very difficult kind of atmosphere, like there was methane, there was volcanoes, there was high temperature. So archaea developed certain features that help them survive in such extreme environments, and some of those organisms still prevails and they survive in extreme environments such as volcanoes. In order to know better about this kind of classification that was done by Carl Voss, let's look into how this classification was done. So as you can see over here, is my slide seen? I want someone to actually respond, or like in audio, like somebody responded. Please respond if my slide is getting seen or not. So Carl Woese was a scientist who basically put forward the three-domain system of biological classification, and this particular classification was done on the basis of the differences in the 16S rRNA genes. So the differences on the conserved sequences in the 16S rRNA gene was responsible for the classification of three-domain system. So as you can see over here, the three domain are bacteria, archaea and eukaryotes. Okay, so from this branch structure you can see that archaea is that category of organisms which show similarity of both features of both bacteria as well as eukaryotes. Okay, so and the similarity of archaea is more towards eukaryotes. Okay, just give me a second, I have to let someone in. Okay, so yes, so what we were discussing was how the features of archaea are almost similar towards eukaryotes, and also some features do show similarity towards bacteria. So as you can see over here, that is why scientists group them more closer to eukaryote as compared to bacteria. So let's see the properties. So the first property of archaea that we will look into is cell membrane. So as you can see, the cell membrane in archaea has got ether-linked lipids. But if you see, for bacteria and eukaryotes, these are ester-linked lipids. So this ether-linked lipids are actually giving the archaea much more strength, or the cell membrane much more strength to survive in the extreme environments where it is still existing. Okay, then the other property, that is cell wall. This property of archaea is similar to that of bacteria. So the cell wall is made up of pseudopeptidoglycan, like peptidoglycan in case of bacteria, and it also has glycoprotein. But in eukaryotes, it can be made of various other structures. The gene structure, archaea is known to have circular chromosomes, just as which perform similar translation and transcription like in the case of eukaryotes. But for bacteria, the circular chromosomes are present, but they have certain unique translation and transcription factors, not as similar as that of eukaryotes. And for eukaryotes, linear chromosomes are present. No circular chromosomes exists, only linear chromosomes are present, and translation and transcription are almost similar to archaea. Just a second, I'll let someone in. Okay, so we were discussing about the three-domain system of classification made by Carl Woese, and we are discussing about how the properties of archaea is similar, or I can say is having a middle ground of properties between bacteria and eukaryotes. But we can also say that the archaea has a inclination of property towards eukaryotes as compared to the bacteria. Okay, so we have discussed till we have discussed the gene structure. Now we will come to internal cell structure. So like bacteria, archaea does not have any membrane bound organelles or nucleus, just like bacteria. But for eukaryotes, there are membrane bound organelles like mitochondria, chloroplast and nucleus. Okay, the metabolism property in archaea includes diazotrophy and methanogenesis, which is only present in archaeal groups, because this metabolism allows it to survive in extreme environments where it can metabolize toxic chemicals for their nutrition. Whereas for bacteria, the metabolism mainly includes photosynthesis if the bacteria is aerobic, and if it is anaerobic, then it includes fermentation. Also for eukaryotes, photosynthesis and cellular respiration, fermentation are major form of metabolism. Reproduction in archaea takes place in the method of asexual reproduction and horizontal gene transfer, which is similar to that of bacteria, but for eukaryotes they mainly participate in sexual and asexual reproduction, not horizontal gene transfer. For protein synthesis initiation, the first amino acid that is formed is methionine in case of archaea, and for bacteria it is formyl methionine, and for eukaryotes it's similar to that of archaea, that is methionine. So here you can see this property of archaea is similar to that of eukaryotes. One second, somebody has to be let in. Okay, so and also the another important factor that we are talking about here is transcription factor or elongation factor 2, which is present in archaea, and scientists have shown that this particular factor is sensitive to a toxin called diphtheria toxin. And similarly, this particular elongation factor in eukaryotes are also sensitive to diphtheria toxin, but for bacteria this particular elongation factor is not resistant to diphtheria toxin. So again another feature where we can see that the properties of archaea and eukaryotes match. Therefore archaea are grouped more closer to eukaryotes as compared to bacteria in this 3-domain classification system. So despite archaea being morphologically similar to bacteria, so as the structure or how the archaea exists may be similar to that of bacteria, but archaea possesses genes and several metabolic pathways that are more closer to eukaryotes, and certain enzymes also they have, like I said, EF2, which are similar to eukaryotes. Therefore archaea is grouped more closer to eukaryotes as compared to bacteria. So the next question here is the primary differences between archaeal and bacterial cell membrane are: A. Bacteria have ester linkages with glycerol and archaea have ether bonds. And the second is bacterial membrane have rings and sugars unlike archaeal membranes. I would like someone to give me the answer, because I just discussed the different features of bacteria and archaea, and the first feature was about cell membrane and the linkages were mentioned. So I want someone to please say what kind of linkages are present in bacteria and what kind of linkages are present in archaea.
Ma'am, option C, A is true and sorry, fourth option A and B both are true.
So you are saying that bacteria have ester linkages and archaea have ether bonds, right?
Yes, ma'am.
No. So actually option A is true. I will show you why. So here you can see the screen is visible, right? So here you can see that the archaeal cell membrane, this is the archaeal cell membrane. You are aware of phospholipid, right? What a phospholipid is. And phospholipid is the monomer of the cell membrane. So phospholipids form together to form the cell membrane, one of the most essential features of cell membrane. Now if you look into this archaeal phospholipid, you will see that this is the glycerol and this is the lipid chain and this is the phosphate group, and the linkage over here is glycerol ether, or we can say that the linkage here is ether bonds. Okay, but Are we seeing any ring structure over here? Are we seeing any ring structure over here? No, right? So sugars or carbohydrates are not present in the phospholipid part of the membrane. So therefore B is not true. But when we are discussing about the archaeal phospholipid, it's better that we compare it with the bacterial or eukaryotic phospholipid also. So over here you can see that the bacterial or eukaryotic phospholipid has got are mainly composed of glycerol ester linkages. Okay, so they have ester linkages in case of bacterial phospholipid, but there is ether linkages or glycerol ether linkages in case of archaeal phospholipid. Now this kind of linkages are very strong and they do not get deformed at high temperatures. So therefore the archaeal phospholipid helps it to survive or helps the archaea to survive in extreme of environments. Also another important thing that you can check over here is that the stereochemistry of the archaeal glycerol moiety is the mirror image of that found in other organisms. So if you check about the glycerol moiety in archaeal phospholipid and that of bacterial or eukaryotic phospholipid, you can see that they are mirror image, and mirror images are called enantiomers. Okay, so since enantiomers are used or made by enzymes adapted for like they are specific, all enantiomers have got a specific enzymes. So this suggests that the archaeal phospholipids or the archaeal membrane that helps in its synthesis of the phospholipids are totally unique as compared to bacteria and eukaryotes, and this kind of enzymes that help in development of this kind of phospholipid was formed in very early when the earth was forming, and gradually this enzyme further developed, and as the evolution progressed, and it developed to form another or develop another enantiomer of the glycerol. Just one second, I have to let someone in. Okay, so we now know that the archaeal phospholipid and bacterial phospholipid are different. So they have archaeal phospholipid over here, they have got a unique glycerol ether lipids, whereas bacterial phospholipid has got glycerol ester lipids. Also the glycerol moiety in archaeal membrane is different, or we can say is the enantiomer of the glycerol moiety in bacterial or eukaryotic phospholipid. This also makes us understand that the enzyme that is used to synthesize this phospholipid in archaeal world is different as compared to the enzyme that forms this phospholipid for bacteria and eukaryotic organisms, and therefore one can say that with time this particular enzyme evolved, and then it got split to form other two domains of enzymes, which forms the other glycerol enantiomer into the bacterial and eukaryotic phospholipids. So now we will get to know how archaeal or how special is the cell membrane assembly for archaeal category as compared to bacteria and eukaryotic category. So you can see that the archaeal lipid tails also differ from the tails of eukaryotes and bacteria. So what is the specialty of this tail? So they are having long isoprenoid chains which has got multiple side branches. So what happens is that when there is multiple side branches, obviously there is more fluidity to the membrane, because these chains will not come together or clump together. If there is more branching, there is more steric hindrance and more departure of this long chain groups, and therefore more fluidity to the archaeal membrane. So this particular feature also helps it to survive in extreme condition. But in contrast, if you see that this fatty acid chains in the membranes of bacteria and eukaryotes are mainly straight chains without much of side branches or rings. Therefore this branch chains in archaea helps it from, or prevents it from lysing or clumping or getting clumped or being deactivated at high temperatures. Okay, so this is another special category of the archaeal cell membrane as compared to bacteria or eukaryotic cell membrane. Also in archaea, the lipid bilayer is replaced by a monolayer. So as you can see over here, in archaea, the tails of the two phospholipid molecules, they fuse together to have two polar heads and in between a hydrophobic region, and this is called Bola amphiphile. This fusion of membranes, or this fusion of long phospholipids to form one phospholipid with two polar groups, makes the membrane of the archaea more rigid, and it helps it to survive further in harsh environments. I'll discuss, but I have to let someone in. Just a second. Now, in order to understand more about archaea, let's see let's discuss about what archaea is. I am sure most of you will know because you have studied in your plus 12 or in your initial stages of college. But archaea is known to be morphologically much more similar to bacteria, and they lack a cell nuclei, or they do not have a definitive nuclei like that of bacteria. So archaea possesses genes and several other metabolic pathways that are more closely related to those of eukaryotes. I have shown in the previous slide that how archaea are much more related to eukaryotes in case of metabolic pathways and certain genes as compared to that of bacteria. Now, one of the most unique feature of archaea is that they are extremophiles. And what does extremophile means? That means they can live in extreme environment like hot springs and salt lakes where generally no other organisms exist. Okay, so if we are to know about certain types of such extremophile archaea, then we have to group such extremophile archaea into four categories. The first category is that of halophiles. So what does halophile means? Halophile means that this category of archaea survive in extremely saline environments such as salt lakes, and the salinity is around 20 to 25%. One example of such halophile is halobacterium. Okay. Other category of extremophilic archaea is that of thermophiles. As the name suggests, thermo means temperature. So this archaea live in high temperatures of 41 to 122 degrees centigrade, and they are found in various geothermally heated regions of earth like hot springs and deep sea hydrothermal vents. Is anybody aware of what a deep sea hydrothermal vent is? anybody aware what a deep sea hydrothermal vent is? Okay, so sorry, I know people are messaging, but I would like more of audio interaction, because as you can see, I have to go back every time to see if people have responded or not. So I would please ask you to put forward audio, or switch on your audio whenever you are wanting to interact. Thank you. Yes, somebody rightly mentioned that a deep sea hydrothermal vent is an under ocean hot spring. Yes, so it is a fissure that is formed inside the seabed, through which geothermally heated water comes out, and this kind of structures are generally found in parts of the earth where there are active volcanoes, and where the tectonic plate of the earth is moving. And this kind of regions actually show how the earth was when it primarily was developing, to form a livable place for us now. So as you can see, from this kind of temperatures, in this kind of extreme temperatures, normal bacteria, fungi or eukaryotes cannot survive. But archaea, because of its unique cell membrane structure, can survive, and one such example of archaea that can survive in extreme heat is Methanopyrus candeleri. The third category is that of alkaliphiles. So as the name suggests, this particular category of archaea survive in high alkaline pH, that is roughly around 8.5 to 11, and this example is Nitromonas pharonis. Acidophiles, as the name suggests, is that they survive in very low pH or highly acidic pH, or such as pH below 2, and this example is Picrophilus torridus. Also, certain of this archaeal bacteria do inhabit the gastrointestinal tract of humans and ruminants. So this also certain number of them assist in digestion. So example of such archaea is methanogens. So as of now, we have known that archaea is one of the class of the three kingdom division of Carl Woese, and archaea has features which are similar to both bacteria and eukaryotes, morphologically more similar to bacteria, and genetically or by metabolically more similar to that of eukaryotes. We also learned how the cellular membrane of archaea is different as compared to that of bacteria and eukarya. We also learned different classes of the archaea, namely halophiles, thermophiles, alkaliphiles, acidophiles, and we learned their example. And also one of the most common archaea that exist in the normal situations, that is methanogens. So now the next question is matching these microbes with dominant domain and habitat. So I would take your time, but I would like someone to come up with the answer, because as of now we know what archaea is and where they stay. So we will discuss about saccharophile and barophile, but since we know about alkalifile and thermophile, I want someone to come up with answer. Yes, anyone? Please respond by audio, because otherwise I have to end the slideshow and again go back. So please respond with audio. Who is it?
Option C. Option C.
Yes. So option C is the right answer. I'm sorry, can I get to know your name? I was just about going back to check your name. Can I know your name?
Neha Maine.
Okay, yeah, right answer, Neha. So the first option is clearly incorrect because it matches psychrophile to archaea. So as of now, let's know, because we haven't discussed about what psychrophile is. Let's discuss about psychrophile. So psychrophiles are organisms that live in that can grow in extremely low temperatures ranging from minus 20 degrees to 20 degrees, okay, and the optimum temperature for their growth is 15 degree. So as of now we did not study that archaea grows in such kind of environment, right? So obviously it is not the right answer. Now matching the psychrophile with bacteria or sea ice, obviously yes, it's matching because certain bacteria are psychrophiles. For example, as you can see over here, one second, I'll just open the pointer options. Like certain bacteria do stay in sea ice or in cold atmosphere, like Anthrobacter, Psychrobacter, and other members of Halomonas, Pseudomonas, and Hypomonas bacteria. So yes, psychrophile is matching with bacteria and sea ice. Now if we go to option 2, that's matching, that has been matched with 4. So let's see, alkaliphile with archaea undersea volcanoes. So in undersea volcanoes, the temperature will be high. Not necessarily it will be alkaliphilic. So obviously this is not matching. Now let's go to option 3, that is 2 is being matched, that alkaliphile is being matched with archaea in soda lakes. Yes, it holds true. We have already discussed that certain archaea are alkaliphiles and they can stay in pH of more than 8 up to 11. Also the fourth option is matching barophile with bacteria in salt lakes. Now let us understand what is barophile. It is an organism that survive under high pressure environment, okay. So this is also a category of extremophile, and the pressure that is persistent is around 380 atmosphere, and this kind of pressure happens in ocean floors and deep lakes, and certain bacteria are known to survive such high pressure. For example, Hanomonas salaria. Okay, this is a gram-negative bacteria that is a barophile, and it can actually stay or withstand a pressure of around 1000 atmospheric pressure. Okay, just wait, I have to let someone in. One second. Okay, I don't think there is pending request. I was getting notifications. Okay, so now we know, we have known about archaea and also other kinds of bacteria which can stay in extreme environment of extremely low temperature and extremely high pressure. Now let's go to the next question, that is, which of these cannot live if exposed to oxygen? First option is obligate aerobic bacteria, second option is obligate anaerobic bacteria, and third option is microaerophiles, and fourth option is facultative anaerobic bacteria. I want someone to answer it. Please take your own time and then answer it.
Obligate anaerobic bacteria.
Yes, true. So let me explain, because I'm sure everybody knows it, but it requires a bit of explanation for some others who do not know. So first let us look at this picture. Okay, is the picture clearly visible to everyone? Is this picture clearly visible to everyone?
Yes, ma'am.
Yes, ma'am.
Okay. So Scientists divided bacteria into 5 categories depending on how sensitive they are to the presence of oxygen and how good they can grow in the presence of oxygen. Okay, now if you look at this tube that has media and that is closed, so obviously this portion of the tube will have maximum oxygen because of the dissolution of oxygen into the media, and this portion of the tube will have the minimum amount of oxygen. Okay, so if we talk about obligate aerobes, we define it as the organism that requires oxygen for its growth. Okay, that is, growth will occur only when there is high concentration of oxygen that has diffused in the medium. So when we keep this organism, or when we grow this organism in this kind of culture media, we see this organism tend to always grow on the upper side of the media. And why so? Why can this grow in the presence of oxygen? Because this particular organisms have specific enzymes. This is the most important part. They have specific enzymes called catalase and superoxide dismutase. This particular enzyme, superoxide dismutase, allows, or it dissociates toxic form of oxygen that has formed, that is H2O2 or any other ROS, reactive oxygen species, that has formed. They neutralize it and breaks it down into H2O and O2, so that this particular reactive oxygen species does not cause any toxic effect in the bacteria. So if a bacteria has to grow in the presence of oxygen, it has to produce two enzymes, that is catalase and superoxide dismutase. So example of this category of bacteria involves Mycobacterium tuberculosis, Pseudomonas and Bacillus. Now let's go to another kind of bacteria, that is facultative anaerobes. So this kind of bacteria can grow in both aerobic and anaerobic condition. Okay, so but if given a chance, this kind of bacteria will prefer to grow in the presence of oxygen. Now as I said, that the concentration of oxygen will be more here and less here. So if you are meant to grow this organism in this kind of culture media, you will see this organism growing more towards the media, more towards where the media is open, and lesser at the bottom. Okay, and this organisms do have the presence of enzymes of catalase and superoxide dismutase, but in lesser quantity this is produced. Example of such bacteria is Staphylococcus, E. coli and Corynebacterium. So as the name suggests, these bacteria can survive in the presence of oxygen. Now the next example is that of obligate anaerobes. So this particular category of bacteria, their growth completely ceases in the presence of oxygen. So if you are meant to grow this organism in this kind of culture media, you will see this bacteria has flocked at the extreme end of the tube. So this bacteria grows only where there is no presence of oxygen. Okay, why so? Because as I mentioned, this bacteria lacks the two important enzymes, that is like superoxide dismutase and catalase, which are responsible for neutralizing the harmful form of reactive oxygen species. And because the reactive oxygen species are not neutralized, so in the presence of oxygen they will react with the bacterial lipids to cause lipid peroxidation, and this will ultimately kill the bacterial cell. Example of this kind of obligate anaerobe is bacterioids, Clostridium, Spastorinium and Methanococcus. Okay, so you can also search for more other examples, but these are the common examples that I have found. Next we will go into the fourth category, that is aerotolerant anaerobes. So if the name suggests, that is, this particular organisms continues to grow in the presence of oxygen, but they mainly prefer the growth in anaerobic condition. Okay, so if you are to see the growth of this organism in this kind of culture media, you will see that they are actually aerotolerant. That means, as the name suggests, they are not restricted by the presence or the absence of oxygen in the culture media. So the growth is evenly distributed inside the culture tube. Okay, so this particular bacteria also has the presence of certain enzymes which allows the catalase and superoxide dismutase, which uh, prevents the formation of the harmful oxygen, reactive oxygen species. But only partially these things are formed, so they can tolerate oxygen to certain level. If you increase the oxygen up to more than a certain level, they will start dying. Microaerophiles are also they require low growth or less concentration of oxygen, and they can survive in oxygen concentration of less than 20%, and they grow where the low concentration of oxygen has diffused in the media. So if you are to check the growth, you will find that they have grown in the position of where optimum dissolution of oxygen has taken place inside the media. This is because in presence of more oxygen, this particular bacteria form does not have enzymes, and they form again the toxic reactive oxygen species which can kill the bacteria. An example of this category is that of Campylobacter. So as of now, we have understood that there are, depending upon the presence of oxygen and how the bacteria uh, uh, can stay in the presence of oxygen, there are 5 categories. That is obligate aerobes, facultative anaerobes, obligate anaerobes, aerotolerant anaerobes, and microaerophiles. Now coming to the next question. So this question I think was present in assignment 0. So the question goes, soybean root nodules are an example of association between a high order organism, plant, and a microbe. What kind of association is this called? Nodulism, parasitic, mutualism, and consortium. Can anybody tell me what kind of uh, association that happens in case of parasitic association?
Nodulism, that is nodulism. The mostly the soybean is having root nodules in which it is going to produce some leg hemoglobin. That leg hemoglobin is going to fix the atmospheric nitrogen.
So actually over here they are saying the association between the plant and the microbe. They are not asking us
Mutualism. Then
about the
mutualism.
phenomenon.
Then
Yes.
mutualism is the right answer.
Yes. So basically, nodulism, we can say that this is a phenomenon, but when we talk about association, we are talking about interaction, how interaction is taking place between two organisms, that is the plant and the microbe. So,
Can we say this mutualism as a symbiosis?
yeah, true. Yeah, that is what I will come into. So the answer is mutualism. Now, because I am I would like to tell how this root nodules actually happen. So once again, I'll go back. I'll just ask. So anybody know what kind of bacteria forms these nodules?
What is the nodule bacteria? Rhizobium.
Yes, right. Rhizobium bacteria is known for forming this kind of root nodule. So what happens is that now by the term mutualism, we understand that one particular organism which are in mutualism with other part of organism, they both are benefiting from each other. This association means that both organisms which are together are benefiting from each other. Now if we are to see this plant is a soybean plant or any leguminous plant, then this bacteria called rhizobium, they grow near the root of the plants, and this particular bacteria forms root legumes or the root nodules, which actually are the region where the bacteria grows inside the plant, and this bacteria actually fixes nitrogen for the plant, where the atmospheric nitrogen gets converted to ammonia, so that the plant can use this nitrogen now for its nutrition. Okay, whereas the bacteria that is surviving inside the nodules are getting the food that is prepared by the plant, and it is surviving inside the plant. So this kind of association that happens where both the bacteria and the plant are benefiting from each other's association, that kind of association is called mutualism. But if the bacteria had only benefited and the plant would have died, then that kind of association would have been called parasitic, which is not the case over here. So the right answer is mutualism. Now if you ask me what is the exact process of the bacterial infection or the process of bacterial nodule formation, so this is a brief, very brief and a sketchy process of how these root nodules are formed. So initially the plant root hair that you can see where there are small unicellular structures, this root hair, they release certain kind of plant hormones like flavonoids, which actually attract the rhizobium bacteria that is present inside the soil. So this bacteria then enters or proliferates into, just a second, I'll check if there's anyone asking to join. Okay, so as I was discussing, one second. So the bacteria that is a rhizobium inside the soil, they sense this particular hormone that is released by the root hair, that is flavonoids, and this rhizobium bacteria then gets attracted to get inside the root hair cell of the plant. So the rhizobium then enters into the root hair and they form a infection thread. Okay, so this infection thread gradually proceeds with the division of the bacteria inside the root hair, and they proceed to go inside the cortical cells over here. So here the infection thread is released and the bacteria becomes spheroids, that is there the infection thread, the membrane disappears and the bacteria develops into bacterioids. This causes the cortical cells of the bacteria to proliferate or to divide, and this division of the bacteria of the cortical cells leads to the formation of root nodules, which actually looks like inflammation, but actually these are compartments for the good bacteria to stay inside the plants. So these nodules are the regions where bacteria are inhabiting, and they are in turn helping the plant to fix nitrogen, that is they are converting the atmospheric nitrogen to ammonia, which can be used by the plant for its nutrition, and in turn the photosynthetic products that the plant is producing is being used by the bacteria. So in general we can say that this kind of association where the plant and the bacteria is benefiting from each other is mutualism. Okay, one second, I'll check if someone else has joined. Okay, now coming to the next question. What are Koch postulates? Can anybody tell me what are Koch postulates?
Option A.
Yes, the correct option is clearly option A. So Koch postulates was postulated by Robert Koch, and it was done in the late 19th century when people were studying and majorly establishing how diseases were formed. So this was one of the primary theory that was given to develop a relationship between pathogen and a disease that the pathogen causes. So he basically listed down the rules that actually shows how a organism or how a microorganism can be related to the disease that they are causing. So the first postulate of Koch was that the microorganism must be found in abundance in all organisms suffering from a disease, but should not be present or found in any healthy organism. So obviously, if a person or if a organism is suffering from a specific kind of disease, like maybe it can be dysentery, then you are expected to find E. coli or excessive growth of E. coli inside the person or inside the fecal disposal of the person, but you are not expecting high dose of E. coli to be present in case of a healthy person. So this is what he said, and that was the first postulate. The second postulate was that the microorganism must be isolated from the diseased organism and can be grown in a pure culture. So he said that microorganism that is causing the disease should be isolated or should be isolated from the diseased organism, and when they are cultured they should form a pure culture. But the same since the healthy organism does not have any microorganism, so no pathogen cannot be extracted from that healthy individual. Next postulate was that the cultured microorganisms should cause disease when introduced into a healthy organism. Now if you have cultured the organism and again if you take out the organism from the culture and further incubate it inside, or you further put it inside a healthy organism, then the healthy organism will develop the disease and become a diseased organism, because the microbe will obviously proliferate inside the diseased organism. Again, if you the fourth postulate states that the microorganisms can again be re-isolated from the diseased individual, and which is actually the experimental host, and it can be found to be identical to the original causative agent. Okay, so basically he has this Koch postulates clearly laid down the relationship between pathogens and specific disease, which are still followed till now. But Koch postulate did have certain disadvantages because they did not take into consideration that the diseases that can be caused by viruses. Okay, and also they did not take into consideration diseases caused by certain bacteria which cannot be grown into artificial media. So he said that every particular microorganisms that are causing a disease can be cultured, but not every virus and not every bacteria that are causing a disease can be cultured. So that was one of the disadvantage of Koch postulate. Also, the second disadvantage was that he did not talk about, or he did not mention about, diseases like pneumonia and nephritis that can be caused by more than one type of microbes. He did not mention about that. He just said that if you are suffering from a particular disease and if a microbe can be extracted from you, again incubated into another organism which is healthy, it will develop the same disease, and if extracted from microorganism is again extracted from that particular healthy or diseased organism, it will show to be the same as the primary one. But in this case, where more than one organism is causing a disease, it might be so that the organism that we have extracted and re-incubated into an individual might not be same as the primary one. Also, it said that the another exception was that certain pathogens like pathogens cause several different diseases. So it is not, certain pathogens does not only cause one type of disease. So the disease that are caused by multiple pathogens or one pathogen causing multiple disease were not considered for Koch postulates. The other was that the pathogens such as HIV cause disease in humans only. So if we are talking about laboratory conditions where we can inoculate the disease into, or we can inoculate the diseased organism into diseased microbe or the pathogen into a undiseased healthy organism, that cannot be done because HIV infects particularly humans. So this postulate was limited to only certain kind of diseases, so it was further modified later. So basically what we understand is that Koch postulate was actually laid down logical steps for proving the relationship between pathogen and a specific disease. Now coming to the next question. Mitochondria is known as the powerhouse of the cell. It is believed that the mitochondria and the independent bacteria but was engulfed by another bacteria. Now mitochondria is an essential part of eukaryotic cell. The reason microbiologists believe this theory is because mitochondria, and the options are, generates energy instead of consuming it, has a tiny membrane separating it from the rest of the organelles, has a unique genetic material, it is where cellular DNA is stored. Can anybody tell me the answer?
That is the
Option
unicellular
A.
DNA.
Can you please repeat?
Unicellular DNA it is having.
Unique cellular DNA.
Yes, it is where the cellular DNA is unique.
You mean option 4?
Yes.
Can anybody else answer?
Option C.
Yes, it has a unique genetic material. Now let's look at a mitochondria, right? If you see, mitochondria has got mitochondrial DNA. Can everybody see the image that I have now shown? The mitochondria, apart from individual genome that is present inside the animal cell or any other eukaryotic cell, there are mitochondrial circular DNA that is existing inside the mitochondria. So mitochondria and chloroplast, similar thing also exists in chloroplast. They have unique genetic material. Therefore, it is believed to be a part of endosymbiotic theory. Now, everybody knows, or anybody knows about endosymbiotic theory? Endosymbiotic theory.
Endosymbiosis is the symbiosis where the living organism will stay within the organism, living organism.
Right. Yes,
That is,
true.
both of them have exchange for either for food or energy or any other needs.
Yes. So endosymbiotic theory basically means it was an evolutionary theory which actually means what you rightly said, that one organism can survive inside that of the other organism, and this particular theory was actually helped the scientist in understanding the evolution of eukaryotic cells from prokaryotic cells. And this theory holds for mitochondria and chloroplast because these are organelles that have double cell membrane layers. So what actually does this theory, or how did this theory come to the scientist? So the evidences that mitochondria and chloroplast arose from a prokaryote or a bacteria is that, first, both of them have double membrane bound organelles. Bacteria and like inside a bacteria you will never find a double membrane bound organelles, but if you look at a bacteria, the bacteria has a cell membrane and cell wall. So scientists said that the one bacteria might have been engulfed by another bacteria, and that led to the formation of mitochondria. Okay, so this is the primary evidence that the mitochondria and chloroplast arose from the bacteria. The next is that this particular mitochondria and chloroplast have their own DNA which are circular. and circular DNA we have also seen to be present in bacteria. Also, the mitochondrial and plastid ribosomes are similar to 70S ribosomes that are present in bacteria and are not present in eukaryotes. So, the ribosome is also similar as compared to that of similar to that of bacteria and not to that of eukaryotes. Also, the mitochondria and plastids are divide by binary fission, which is similar to that of bacteria. Also, the proteins that are created by mitochondria and chloroplast during transcription, the initial protein is N-formylmethionine, which is actually similar to that of bacteria and not similar to that of eukaryotes. So we can clearly from here we can say that mitochondria was actually a bacteria that was engulfed by another cell, and that then got a specific job, that was to do electron transport chain or to generate energy. Okay, similarly chloroplast was a bacteria that had the capability to produce food in the presence of sunlight, but that bacteria was engulfed by a cell, and then this particular chloroplast was formed, which then is essential for the cell to produce food or to perform photosynthesis. So what I said can be further described by this particular diagram. So now if we are talking about origin of mitochondria, so scientists believed that mitochondria basically came from an ancestral anaerobic predator cell. So this was the predator cell which was anaerobic, that is they could not survive in the presence of oxygen, and this particular anaerobic ancestor, they engulfed the bacteria which they engulfed the bacteria which was basically an aerobic bacteria. So this aerobic bacteria means that they can perform the electron transport chain to generate energy. So this bacterial cell engulfed the aerobic bacterial cell, and gradually the bacterial cell lost membrane, and they finally stayed inside the particular predator bacteria to generate or to evolve into a eukaryotic cell, where this particular bacteria that was engulfed became characterized by producing energy for the cell, and therefore it was called powerhouse of the cell. So this kind of uh, relationship that existed between the predator cell and the bacterial cell that was engulfed was symbiotic relationship, wherein this small bacteria was staying inside the bigger predator but was not getting killed, and it was staying in the exchange of certain metabolites and certain form of energy. So this is how mitochondria developed. Now similarly, if we have to know the origin of chloroplast, so it is believed that an early eukaryotic cell which already had the mitochondria engulfed a photosynthetic bacteria, or the bacteria that can produce that can produce food in the presence of sunlight and carbon dioxide. So when this bacteria was engulfed, it again became an endosymbiotic relationship, and then it developed into formerly known plant cell or eukaryotic cell, which has both mitochondria and chloroplast. Now coming to the last question of today's session, that is, which of the following can be used for isolating bacteria with a target gene? The first option is heterotrophic plate count, second option is biological oxygen demand, third option is selective screening, and fourth is dilution to extinction. Yes, anybody answered?
B, madam.
Yes, sorry, I cannot hear you.
Selective screening.
Selective
Yes.
screening.
So, can anybody tell me what is selective screening?
It is
It is
selective screening
selective
is
screening is
separately
separately
we'll
will
select a
select
particular
a
gene
particular
and
gene
target
and
genes
target
and
genes and
based on that
based
we
on that
can
we can
in the
in
media
the
we
media
can
we can
cultivate that
cultivate that
a
a
specific
specific
bacteria with a
bacteria with a
particular gene,
particular gene,
like genetic
like
Yes.
genetic engineering,
where we
where we
inoculate a part
inoculate
of
a
the
part
gene
of
in
the
the
gene in the
bacterial gene and
bacterial gene,
we'll
and we'll
culture that in the
culture that
tissue
in the
culture
tissue culture
and use
and
Yes.
that
use
screen use that for
use that
screening
for
in
screening in
SDS
Yes.
page.
SDS page.
So basically, when we are talking about a target gene and when we are talking about a bacteria, we are basically talking about recombinant plasmid, right? When we are, and this is basically the basics of recombinant DNA technology. Just one second, I'll just check if anybody else has joined. Okay, so when we are talking about a target gene and a bacteria, we are basically talking about recombinant DNA technology. So if we are aware that in recombinant DNA technology, we have a target gene, that is a gene of our interest. It can be anything. It can be an insulin gene which we want to produce inside a bacterial cell or overproduce inside a bacterial cell to give us a target protein. Okay, so what does one do, or what does a scientist do when they want to do that? They take a plasmid that is from a bacteria, they cut the gene, they cut the target gene with restriction enzymes, and they then ligate the gene with DNA ligase into a site where multiple restriction sites are present, and this particular plasmid that is now produced is called recombinant DNA plasmid. Okay, so now in order to produce or make more copies of this plasmid or more copies of the protein of interest, we require a medium or a biological organism which can form protein from this particular gene. So for that we need to transform a bacteria with this plasmid. Okay, so now once the bacteria is transformed, it is called a recombinant bacteria. So before we delve into what this is and what selective screening is and its example, let's talk about the other options that were given. So the first option was heterotrophic plate count. So as the name suggests, heterotrophic plate count is basically the standard plate count technique. So suppose you have taken a sample from pond or from oil or from soils. Okay, so what do you do with that in order to extract the bacteria that is present in the water, pond water or soil? What we do is we make serial dilutions of that concentrated sample. So each of these dilutions that we are making are then poured over an agar plate, and it is spread plated to actually see how much colonies are being developed and if we are getting the bacteria that we were looking for. But obviously here no one is taking into account a target gene, or nobody is taking into account the gene of interest that we are talking about, and there is no transformation of bacteria, no insertion of recombinant plasmid taking place. So obviously this is not the answer, because here we are not dealing with a target gene. We might be dealing with a selecting a particular bacteria from the soil or from the like different atmosphere from where we have collected the sample, but definitely not this is not a good method to check or to isolate bacteria with a target gene. Now coming to the next option, that is biological oxygen demand. Anybody is aware what a biological oxygen demand means?
The
The
oxygen
oxygen
required
required
to
to
complete its
complete its
biological
biological
activity.
activity.
The amount of
The amount
oxygen
of
required
oxygen
to
required to
complete the
complete the
biological
biological
activity of
activity
a
of a
particular
particular
bacteria.
bacteria.
Is it correct?
Is
Um,
it correct?
Actually, biological oxygen demand is basically the amount of oxygen that a microorganism will require to break down organic materials in water. So basically, this is a category, or this is a the characteristics that by which scientists measure if the water is polluted or not. So obviously, if the water is clear or does not have much of organic pollutants, then the biological oxygen demand will be high, obviously, because less oxygen will be used by the bacteria to break down such organic materials. Okay, so as you can see over here, this is microorganisms break down excess organic materials. They use up dissolved oxygen and deplete the oxygen levels. So the more you have organic materials means less of the oxygen inside the water. So the this biological oxygen demand therefore shows us if the bacteria is contaminated by organic materials or not. So the lesser the oxygen, the more the bacterial presence. So because the bacteria is using up the oxygen to degrade the organic impurities present in the water. So clearly, whatever we are learning or understanding now, this cannot be a representative for understanding or isolating a bacteria with a target gene. This is just a this biological oxygen demand can just help us to characterize a particular solution that is given to us. So obviously this option is also not true. Now coming to the third option, that is selected screening. Yes, this option is the right answer, and I will end the discussion by actually talking about an example of such selective screening. So anybody aware of blue white screening?
No.
Okay. Anybody else? Are you aware of blue white screening?
There is one
There is
other
one other
example for
example for
gene I
gene
know.
I
What
know.
is that?
What
Is
is that? Is
the
the
to kill
to kill
the some insect
some
pests
insect pests
will
will
inoculate a
inoculate
particular
a
gene
particular
called
gene called
Bacillus
Bacillus
thuringiensis gene.
thuringiensis gene.
Yes, Bacillus thuringiensis gene actually can you can call it a target gene, but it is. But can you call it a like a selective screening procedure?
Bacillus
Bacillus
thuringiensis will be
thuringiensis will be
firstly
That
select
is
that is
that
selected
is
in
selected in
selective screening
selective
only. Then
screening
it is
only, then
going
it
to
is
be
going to be
incorporated in the
incorporated in
seeds.
the seeds.
yes. So no, actually, by talking about selective screening, I just wanted to know about like how scientists choose if a particular cell has been transferred, or if a particular bacterial cell has been transformed or not. How that is being understood. So that is what I meant. So
I don't know.
I
Actually,
don't
it
know.
might
Actually, it might
be maybe
be
due to
maybe due to
fluorescence
fluorescence
dying or
dying or
like that. I
like
don't know
that. I don't
actually.
know actually.
yes. So I'll be explaining that here. So I was talking about plasmid in the previous slide, right over here, which is basically the vector that holds our target gene. Okay. So generally a plasmid has certain different features. The first feature that it has is multiple restriction enzyme site where we can put our gene of interest, because this region can be cut by different restriction enzymes. The second thing that it has is a antibiotic resistance gene. Now this antibiotic resistance gene, the presence of this gene can serve as a selection marker. So if a bacteria has this plasmid, it will be resistant to this particular antibiotic. Here in this case it is ampicillin resistant. Okay, so now if you look at this picture carefully, this bacteria has got different copies of the plasmid, which has the antibiotic resistant gene, and also it has the gene of interest, which is marked by this red part. So since it is exhibiting this antibiotic resistant gene and the corresponding protein, so it can stay alive even in the presence of antibiotic, which we are putting in the media where we are growing the cells. But the bacteria which is not transformed by the plasmid, or which has not got the plasmid or the gene of interest, will obviously die. So if we are able to grow the bacteria uh, that has got the plasmid, we are sort of close to isolate bacteria that has got our gene of interest. But is it that easy? No, it is not that easy. When we are working with this kind of uh, plasmids, there is a chance of self-ligation. What does this mean? This means that suppose you have cleaved this plasmid with restriction enzymes, okay, and you have then put it in the presence of a target gene. Sometimes it might so happen that the target gene did not get incorporated, but the plasmid itself got ligated with its sticky ends. Okay, this can be a scenario. Now there can be a chance that this self-ligated plasmid and this plasmid having our gene of interest both exist in the medium with which we are transforming the bacteria. So there is a chance that the bacteria can get a plasmid which does not have our gene of interest. Now in that case, the bacteria will still grow in an antibiotic in a plate that has got an antibiotic because it has become resistant because of the presence of plasmid. But that doesn't necessarily mean that the bacteria is having our gene of interest. So this is where blue-white screening or selective screening come into place, where we can selectively target and selectively pinpoint towards those bacteria which has our gene of interest. Okay, so blue-white screening is a efficient technique which helps us to understand recombinant bacteria, and the main understanding of this technique is that it relies on the activity of beta-galactosidase, that is an enzyme that is present in E. coli, and it cleaves lactose to form glucose and galactose. So this enzyme is the primary concern of blue-white screening. So what happens is that first let's come towards the plasmid that is there. So the plasmid over here, you can see there, this is lac Z or lac operon that is present, and there is a yellow colored part which shows the site where the gene of interest can be put in. Now when this part is not cleaved or this part does not have our gene of interest, then a stable beta-galactosidase protein is produced from the transcription of this part or translation of this part. Okay, this beta-galactosidase, once it is produced, they actually break down X-gal, which is a media component that is present in the agar, that is present in the agar where the bacteria is growing, and it hydrolyzes it to form beta bromo 4 chloroindoxyl, which then dimerize to form a blue pigment which is called 5,5 dibromo 4,4 dichloroindigo, which gives blue color. Now this blue color being produced, the bacteria wherever it is growing, it will show a bluish colored colonies. So if I have to repeat it again, those plasmid which does not have a gene of interest will be successfully able to produce beta-galactosidase enzyme, and this beta-galactosidase enzyme will break down a synthetic form of galactose, that is X-gal, which is already present in the agar medium where the bacteria is being growing, and it will produce or hydrolyze to form 5-bromo 5-chloroindoxyl. This 5-bromo 4-chloroindoxyl will then dimerize to produce a blue pigment which is called 5,5 dibromo 4,4 dichloroindigo. So this blue coloration will be formed by the colonies which did not receive our gene of interest. Now if we look at the insertion of our gene of interest inside the plasmid, then you can see that it has separated separated the portion of the plasmid that was producing the beta-galactosidase enzyme. So after transcription, no functional beta-galactosidase will be produced. Since beta-galactosidase will not be produced, therefore the there will be no generation of the blue pigment, so the bacteria that are recombinant will be growing to produce white colonies. So if you are growing this kind of bacteria on Petri plates, you will be clearly seeing both the presence of blue and white colonies. So the blue colonies means that your gene of interest has not been incorporated into the bacteria. Though the bacteria got the plasmid, it got this plasmid which was self ligated and not the plasmid that had our gene of interest. But if you are having or seeing white colored colonies, then you are sure that the bacteria has received its gene of interest, and therefore they are not able to produce any functional beta galactosidase enzyme, and therefore the colonies that are growing up, they are appearing as white because the dissociation of X-gal is not taking place in the media. Is there any doubt regarding this particular blue-white screening?
Yes, we understood, but can we get notes of this? Where we can get the notes of all these lectures?
So actually I will share in the next class, I will share the link of the YouTube link of these videos. These videos will be uploaded in YouTube, so you can get the notes, and I will also give the slides link.
Thank you.
Yeah.
Thank you, ma'am.
Okay. Okay, so with this I end my presentation. I would be happy to take questions if there are any. Is there any questions?
In second, in the second week, we're facing a little bit of problem because lot of bioenergetics in this. Most of the calculations, they are biology students. We don't know mathematics. That is.
Okay. So actually in this particular live session, it was mainly targeted to discuss the initial week's assignment. So as the live sessions progresses, we will discuss about the bioenergetics part also.
Thank you. And in this we don't have any doubts. For myself, I don't have any doubts. Thank you.
Okay. Thank you.
Thank you.
If nobody has doubt, then we can end the session over here. We will meet again on next Tuesday at 7 to 9.